for now, what I wanted to do is basically give an overview of, from one person's perspective of what is oncology drug discovery, and in particular, how our group goes about doing it. So I'll start off with a slide that has been shown previously, but basically it's a mission of our group, and that is that we're here to discover, take cancer discoveries, be them from local academics, from entrepreneurs, from the OICR network, which includes people in this room, and take these studies and do translational research to find chemical probes and leads and ultimately clinical candidates. And one of the themes I want to build on here is that, ideally, at the end of the day, we want to benefit the lives of cancer patients, but our group is too small to be able to do it ourselves. We absolutely have to rely on the help of Facet, who identifies strategic partners for us so that we can not only get the funding, but also the capabilities to go into more advanced phases of drug discovery research, which I'll get into. And so, a word about who we are. Well, we've been growing over the years. We're now comprised of about 40 scientists, and some of them are students, but many of us are veterans from the pharmaceutical industry. We come from various different companies as well as straight out of school, and we have different capabilities. And so our group got started around 10 years ago under the direction of Rima Alawar. And really the emphasis was on medicinal chemistry, compound libraries, and computational chemistry. And over the last few years, we've really augmented that capability with an ever-growing group of biologists. And these biologists do biochemistry and cell biology, and they do assay development work. And really, that group has been instrumental in helping us leverage our medicinal chemistry capabilities and working with academic people to help design screens and test compounds and validate targets. And then all along, we've had an excellent capability in terms of our analytical group and being able to develop studies for ADME, which stands for absorption, distribution, metabolism, and excretion, properties of our drugs, and also in vivo pharmacokinetics, which is an essential portion of drug discovery. And so, that's who we are, and essentially, what are we trying to do? Well, in this slide, I've tried to depict really a grossly oversimplified view of the three main categories of cancer drugs. And they're basically, if you start on the upper left, it's cytotoxic agents, and those are your typical classical chemotherapeutic agents, tubulin binders like Taxol, DNA intercalating agents, and other traditional chemotherapeutics. All of these can be very effective because they kill cancer cells, but they also kill normal cells. Therefore, they come with a lot of side effects. They are still a mainstay of cancer treatment, but in combination with other agents. And so the two emerging areas, one is quite well established. It's targeted therapies at the bottom, and that's been an area that's under ripe exploration for the last 20 years. And these include things like small molecule kinase inhibitors, anti-apoptotic agents, and various targeted antibodies, such as trastuzumab for breast cancer. And there's a very exciting emerging targets that target PPIs, which stand for protein-protein interactions. And there's also antibody-drug conjugates. And so why that's highlighted in blue is I think that's an area of particular interest and particular emphasis in our group. And finally, on the upper right, everybody who's involved with cancer research knows about immunotherapies, immuno-oncology, and I'll get into that more later, but of course the mainstays are PD-1 blockers and CAR-T, and then there's very exciting emerging data around oncolytic viruses. And so that's the overall space, and you can also subdivide cancer treatments into two therapeutic modalities. The first being biologics, which basically are proteins that are produced in settings outside of our labs. And then there's the small molecules, and there's a lot of comparison and contrast that can be made between these two modalities. but I think it's fair to say that they each offer their own advantages and disadvantages. Biologics, of course, are very specific, very targeted, and when they're injected intravenously, they actually can stay in the body for quite a long period of time and can be very, very highly effective. On the other end, small molecules are able to be taken by pills, and they're synthetic compounds. And really, I think there's different settings for different approaches. Where our group generally plays is in the area of small molecules. Although I will say we do have an interesting and exciting project with the antibody drug conjugates, which is in a way a marriage of the small molecule and the biologics when you're attaching a small molecule to a biologics to make it more targeted. And I don't have time to go into that approach in any more detail than that. So here's just a general view of drug discovery and development, and many of you have probably seen this or similar types of depictions of drug discovery. And really the whole point here is to emphasize that it's a very long, laborious process that's fraught with risk. You start out on the left with a target that's being validated. You have to do the initial validation work, and then you have to figure out how to drug the target, and then make a better drug and a better drug, and finally you go into advanced preclinical studies, and then you go into the clinic. And so our group is really just involved with the very early stages of this. When we're successful, as we look forward to being in the near future in terms of taking compounds into the clinic, we're going to want to be partnered with other organizations who have the expertise to be able to do that. And sometimes from the point of view of a person in drug discovery, we feel a little bit like the guy in the foreground who's facing a daunting exercise of taking a small molecule and optimizing it to go into the clinic. And this is something that we're always aware of. I think what I like to tell people is often we get discouraged looking at the end point when in fact we have to look at the intermediate milestones along the way and take care of those intermediate hurdles before we achieve our ultimate success. Because after all, that the big diagram that I showed earlier usually takes about 10 years from the time when you discover a validated target to when you actually develop the drug and the drug actually reaches the marketplace. So it takes a lot of patience and a lot of fortitude to be able to go through that process. And so in this scheme of things, what I wanted to emphasize is not necessarily as much the target identification and validation, because I think that's a topic that's better treated through the biologist and the biology expertise, but really from a chemocentric point of view, how we actually go about prosecuting validated targets. And so when I say a validated target, that can mean a lot of different things. It could be a target that's of interest from the standpoint that there's some preliminary literature data. There could be some genetic data that comes out of the clinic that's pointing to that target that then makes it interesting. There's a lot of different ways that something can be validated, but the point is that once it is achieving a level of initial interest, that's when the medicinal chemistry team goes about trying to investigate that target. And one of the earliest things we do is to run high throughput screens on a collection of compounds, and then we'll get a hit. And that hit has to be confirmed by retesting, usually from starting from a liquid set of liquid compounds to retesting the powder. And then a lot of these hits then are looked at by the medicinal chemists. We decide which ones look most promising, and then we start making analogs of those particular hits to figure out which one looks the best. And then after we take a hit that looks good and it's basically been confirmed, then we start to do more labor-intensive medicinal chemistry optimization on that hit. And what we're looking for is to basically improve the properties of the hit, make it into something that has better potency, has good drug-like properties in terms of permeability. It has the ability to show good stability and good solubility, and really can start to reach the biological target of interest. And so after continuing to do the lead optimization, we have a more advanced compound that is then chosen for advanced studies that are often more costly. Usually they involve animal work in terms of actually doing significant efficacy, PK, and things of that nature, and that's where we get really excited, and we're very focused on putting all of our resources into the most promising area. And so once a molecule is really pushed into the clinic, of course, we want to ask key questions. And really, the earlier we ask these questions, the better, even though they're somewhat in the domain of the clinician. If we have an appreciation for these questions very early on, we're much more likely in the drug discovery group to be successful. And so questions that are always raised are, does the patient population express the target? And you have to make sure that, as obvious as that sounds, many drugs are taken into the clinic against patients who have no chance of benefiting because they're not the ones who are actually expressing the target of interest. And does the compound show acceptable pharmacokinetics? That's something that's relatively easily measured in the clinic, but we're getting better and better at actually predicting how good the PK is going to be in humans using preclinical models. Does the compound engage the biological target of interest? And is it well tolerated? And finally, is there an early hint of efficacy? And the sooner we see that, the better. And so how do we actually go about doing that in the preclinical in systems? Well, here's a simplified drug discovery cascade. And really what our group is all about is taking larger numbers of compounds that have either been obtained through compound collections or synthesized, and filtering out the best ones for more advanced studies and taking them as potential clinical candidates. And so this is kind of a simplified project cascade, and the reality is one that looks a little bit more complicated. And it's basically, we take compounds that we synthesize or obtain, we study their activity through enzyme activity, their biochemical activity, and then we take them into cell-based assays, and then into more and more advanced and complicated assays such as PK and in vivo efficacy. And so now what I'd like to do is to try to illustrate how that worked with one particular project. And before I do so, I wanted to make sure that I highlighted that it all starts with compound libraries, and the quality of our libraries that we have really helps dictate our success, because if we don't have the proper starting points, then we can't really get anywhere. And so there's two main types of libraries. There's a high throughput screening library, which is comprised of many compounds, and it's amenable to screening under formats which can go through tens and hundreds of thousands of compounds in a matter of weeks. And we try to get as much chemical diversity in there, and we try to get examples of compounds that are going to hit as many different types of biological targets as possible. The OICR Drug Discovery Group has a compound collection for their high throughput screening of about 140,000 compounds, and we're still trying to build on that. Meanwhile, we have various targeted libraries, and a targeted library is loosely defined as one that is designed around a particular set of targets in mind, for example, kinase inhibitors or protein-protein interactions. And usually these groups of compounds is much smaller, and it's amenable to types of assays which are less high throughput. And so there's different approaches in terms of how you use these types of libraries, but I think it's fair to say that most of our efforts are around trying to find these specific biological targets and screening these libraries against those biological targets. But as this slide represents, there's another very interesting approach, which is called phenotypic-based screening. And so that's where you screen molecules in a cellular format, and you don't necessarily know what the type of target is that's actually causing the effect, but you're looking for the effect, and then you can actually work your way backwards or deconvolute to figure out what the biological target is. And so I won't get into that approach too much in this talk. What I'd like to do instead is focus on the target-based approach and use one of the projects in our group, WDR5, as an example. And so WDR5 was a very interesting target that was actually brought to our attention through the SGC, or the Structured Genomics Consortium, who's right down the stairs from where most of the MedChem group is, on the 7th floor of the South Tower, and partly in the 6th floor as well. And we had begun working with this group because WDR5 looked like an interesting target from a standpoint of having an effect in epigenetics. And epigenetics is basically the looking at the changes in that affect the genome without actually changing the sequence of DNA itself. And with WDR5, not to get into the biology of this target too much, but just to show that it's actually part of a very important and complex structural complex comprised of several different types of proteins. It's called the compass complex, and one of the many things it does is actually methylates or puts a methyl group on histones, which then leads to changes in the expression of DNA. And so it's been shown through a variety of literature evidence that that may have a role in cancer. And I will say, though, that going into this project, we weren't fully appreciative of how much of a role it had. And so what we did initially, and this was a part of the early collaboration with the Structured Genomics group, is we screened a subset of our high throughput compound library. It's called a 16K diversity library, using an assay called the fluorescence polarization assay. And then we reconfirmed hits, and we selected the most promising hits for future work. And so the hit that's shown on the right-hand slide here was actually one that emerged from this screening. And in the grand scheme of things, 12 micromolar is not nearly potent enough to be considered a drug, but it was a very potent for an early hit. And so it looked like a very promising scaffold as well to be able to do more modifications of it. And so this part of this early collaboration was actually confirming, and this was a critical part of the step in the process, was confirming that it actually bound to the biological target, which is a portion of the WDR5 protein. And it did so in a way that made a lot of sense, and it displaced the endogenous ligand, or the partner of this protein, in a way that we thought maybe this could be efficacious. And so then further work was done on this hit, and this was all part of that early collaboration. And we did what's called SAR, Structure Activity Relationship Investigations, and within just a few years, this hit was turned into a probe. And the probe is called 9429. And what is a probe? Well, a probe is something defined by SGC to basically mean a molecule which is potent and specific for that target, and can be used by the scientific community at large in order to help understand biology more. And so that was an exciting part of this project. But what the Drug Discovery Group then decided to do is to build on those early investigations, which were published, and to develop new proprietary molecules that actually had further expands the value of this initial finding. And so there was two aspects to this. One was to make a probe that was so-called second generational probe, which is more potent. And the reason for doing that is because even though 9429, the original probe, had good biochemical activity, it wasn't really so spectacularly potent in cells. And so the idea behind having a second generation probe that's more potent, maybe you can further understand and delineate the biology and show the potential. And of course, in the lower right is another area that we were very interested in, really getting back to the heart of the mandate of our group, which is to discover a clinical candidate drug which could be taken on and partnered with various organizations to fully realize the clinical and therapeutic potential of this target. And so a lot of steps involved in there, but what our group decided to do was basically take the probe, and what typically medicinal chemists like to do is they like to break up a molecule into sections, or so-called zones, and make different chemical changes to do so. And then our goals were really to further validate the biological target and to advance the drug into a clinic, as I said earlier. And so what we did is through a lot of medicinal chemistry, SAR, we came up with this lead molecule 11824. And, you know, one of the key points here is that we went from initial probe, which was already quite potent, of around 30 nanomolar, to one which was at least 30 times more potent, called 11824. And so that was really considered our lead for further efforts to go on and develop a clinical candidate, which we think we're well on our way to doing. And stay tuned. Hopefully we'll be able to provide more details in that respect within the next little while. And so here's the overall goal. And so one of the things that 11824 did, which really demonstrated itself as a lead, is it actually had outstanding cellular activity. And the left-hand chart basically shows, or graph shows, its ability to inhibit the methylation of a histone H3K4. And you can see what is striking about that is that the original probe molecule, which is published, 9429, had only marginal activity in cells, whereas 11824 and a very closely related compound, 12003, actually had a nice sigmoidal curve in terms of inhibiting the methylation. And that was actually great news because it also recapitulated what we found from genetic knockdown studies using shRNA. You can see that actually the small molecule was behaving very much like those knockdown studies, and that actually is a good example of target validation when you know that your molecule is working the way the genetic studies indicate it should. And so we went on, and our ADME group got involved, our analytical chemists, and looked at this compound and actually did some studies to show that 11824 was permeable to a somewhat of a degree that was enough to maybe show that it could have some potential, but it wasn't super permeable, and that was definitely an area where we could further optimize. But at this point, what they did demonstrate is that the compound was quite stable under liver microsomes, and that suggested that once you could get it into the organism or the body, it would stick around long enough to have an effect. And so having that initial promising data, what was then done was to take this molecule into mice and show that it actually had decent exposure. And interestingly, as you dose up higher, you get significantly more exposure. But the good news here is that the amount of drug that was able to, through oral administration, be found in the blood vessels of the mice was significantly higher, at least 4 times as much as 30 times higher than what you would see in tissue culture with the inhibition of the cell lines. And so that's a place where you really want to be in a drug discovery program where you actually know that you can inhibit the target in the body in an in vivo setting. And so that's really what led us to take this compound into more advanced studies. And these are so-called in vivo efficacy studies, and the way they run is basically taking mice which are immunocompromised, which are injected with a human tumor, which would typically grow unchecked without some kind of drug, and that's represented by the black line there in the graph. And what you can see here, which got us really excited, was the fact that 11824 was dose-dependently reducing the growth of the tumor. In fact, at some points with the highest dose, you could even see tumor regression from the initial size of the tumor, which was very gratifying to see. Also, because sometimes you can kill a tumor cell, but it's not without a huge amount of toxicity. In this case, we were very pleased to see the fact that we saw the in vivo efficacy without a huge body weight change. And one of the things I want to point out in both of these graphs is the fact that the red diamonds are cytarabine, which is a known clinical standard for AML, which is the tumor type that's being treated here. And you can see that cytarabine actually has significantly more toxicity than our molecule. So this was the kind of data that really, I think, put the program on the fast track, and it allowed Facet to go out and really highlight how much exciting progress had been made. And so, oh, I forgot to say it before I go into that. Just one of the things that we always want to see in an in vivo efficacy study is, in addition to this tumor growth, you want to actually have some evidence that's working by the mechanism that it's supposed to be working at. And in this case, it was because we'd done, the biologists had done some preliminary early studies that showed which genes were knocked down when you inhibited WDR5, and those very same genes were knocked down in the tumor in an in vivo setting. So all of this was the type of data we needed to arm Facet with you know, some exciting pitches to various organizations, and we went out and they actually met with many companies, and we did as well, to try to highlight and showcase the value of this program. And ultimately, we were able to secure a deal with Triphase, which is, as most people know, is an Ontario-based accelerator who's working very closely with Celgene. And we made a deal which, if fully realized and the drug goes into the clinic, could be worth up to $1 billion. But upfront, at least it was worth $40 million, and that's the kind of money that really helps us keep going, finding the next generation of new targets, and it helps Facet in terms of being able to fund this. And I just want to highlight Facet's role in this as well, because really Facet took the risk of this project and helped fund it well before we made this deal. And so they were basically investing in the future, and I'd like to think the investment paid off. And so with that, just want to try to see. I don't know why it doesn't advance. I guess it's because. I think it says a new version of Java.
Okay.
Oh, I guess I have to hit escape.
Yeah.
And then. Perfect. Thank you. And so, as I said earlier, we've got a lot of exciting new projects going on, and just recalling back that earlier slide I showed with the three main therapeutic areas, one of the key ones that we're interested in now, and at least half of our group is now involved with, beyond the success, is to try to reproduce the success of WDR5, but with different targets. And immuno-oncology is obviously the hot area these days, but it needs to be reinforced that immune checkpoint inhibitors, which are the biggest drug in cancer out there, they're making the biggest splash. Even though they're very exciting, they only work in a minority of patients, I should say. And many of these, even if it does work initially, their tumors do come back. So there's huge room for improvement. I think most of our efforts are around how do we improve those types of drugs, not necessarily replace them, but improve upon them in combination therapy. So with that, I can finish up and just show the team that was involved with the early probe work. This is a combination, as I said, of OICR members and SGC. And then this is our drug discovery team. Actually, this is an outdated slide. We're going to have another slide taken on Wednesday. I apologize for those of you who are not in this slide. And then there's people in this slide who have gone on to other things. But this is the great group. It's a lot of fun to work with these people, and I feel incredibly honored to represent them in this particular talk. So thanks very much for your sticking around.